Hello, good morning, my viewers. I hope you guys are well. Today, we are going to have a discussion on antibiotics. I'm going to make that topic to be very simple to understand. But before we look at a summary of antibiotics, I want us to have a brief reminder on what we are to discuss today. So, if you hear the term antibiotic, these are drugs that are used to either stop the growth of a bacteria, bacteriostatic, or to kill a bacteria, bactericidal. So the drugs that are used that work against the bacteria are antibiotics. But before we go to the classification of antibiotics, you have to have an understanding of how a bacteria looks like or how a bacteria works. So basically, you have to understand the anatomy and physiology of a bacteria. And this we always do in a unit called microbiology. In microbiology, we study virology, we study bacteriology, we study fungi in its whole class, and others. So in bacteriology, we are able to understand how a bacteria looks like and how it works. So once you have the understanding of how a bacteria looks like and how a bacteria functions, then it will become easy for you to understand the mechanism of action of antibiotics. Yeah. For instance, sorry to say this, if you want to kill an animal or kill a human being, sorry to say this, you have to understand how the animal works or how the animal looks and how to destroy that animal. So same applies to a bacteria. You have to understand how a bacteria looks like and how it works for you to be able to stop it. So in this class, I'm just going to brush through bacteriology. I'm not going to go deep in it because the main aim of this class is to have a clear understanding of antibiotics. So I'm just going to brush through bacteriology just to remind us on the basics that you have to know in order for you to understand how the antibiotics work. Now, in the screen below, if I zoom it a little bit, you can see as a picture. This is a structure of a bacterium, yeah? So that these three components that I want you to understand or I want you to have a clear understanding on. And this is bacteria as a whole. There are three key components that you have to have an understanding in. So the bacteria is very similar to a human cell in that it contains the nucleus or the nucleoid, as you can see in the diagram below, a nucleoid. The bacteria also contains a cell membrane, but the only difference is that it has a cell wall. So in this diagram or in a clear picture, you have to have a good understanding on how the cell wall works in a bacteria, on how a cell membrane functions or looks like, and finally how the nucleus works. So basically, the drugs that destroy bacteria by acting on the cell wall or acting on the cell membrane or acting on the nucleus as a whole. And remember, nucleus contains all those, the DNA, the RNAs, and all those things that compose the nucleus as a whole. So you have to have those the understanding of those three foundations: the cell wall, the cell membrane, and the nucleus. Yeah. So as you see this diagram here, we've got the cell wall. The cell wall is majorly composed of peptidoglycan layer. Cell wall is majorly composed of the peptidoglycan layer. And one thing that you have to know, peptidoglycan layer basically is like a rigid polymer. It forms a structural component of most bacterial cell wall. It basically provides the mechanical strength and protects the cell from bursting because it has got high internal osmotic pressure. So it's a very rigid, strong mechanical structure that ensures the bacteria is well protected. Yeah. So this is the cell wall, and this diagram, if you see color red, This is the cell wall. From this index here, you can see the cell wall is red in color. So that is the cell wall. Underneath it, you have got the cell membrane. Some call it cytoplasmic membrane. Yeah. I'm going to give major difference if you look at the classification of the bacteria. Some have got, you've got also the capsule, you've got the nucleoid and other structure, the flagellum, you've got the ribosomes inside, and other organelles very similar to the human cell. Now, if you look at uh, bacteria as a whole, we have got a very two broad category. We have got the gram-positive bacteria and the gram-negative bacteria. So, in your understanding of the antibiotics, there's some drugs that are very convenient against gram-positive and some that are very convenient against gram-negative. So you have to understand why this happens or why this occurs. For you to understand how this happens, you have to understand the anatomical difference between the gram-positive bacteria and the gram-negative bacteria. So the one common difference is on the cell wall. So in gram-positive, the cell wall is always thick. So here we can put it always contains a very thick cell wall. Some people can say it contains a very thick peptidoglycan. Why? Because peptidoglycan is what constitutes the cell wall of a bacteria. So till there, I believe we are together. So in gram-negative is the opposite. It contains a thin cell wall, or you can say a thin peptidoglycan. So one key difference between the two is that if you look at the gram-positive bacteria, the cell wall is extremely thick. Why? Because the peptidoglycan layer, which constitutes the cell wall, is thick. Whereas in the gram-negative, the peptidoglycan is thin. Another key difference is the outer membrane. So here is where the difference comes in. Now we always have a cell membrane. You always have a cytoplasmic membrane. So we have got an outer and inner. Now, one major difference is that in gram-negative, they always contain an outer membrane. So this is always present. Yeah, and this is always absent. They lack an outer membrane. And this being said, the outer membrane is always composed of lipopolysaccharide, lipid and polysaccharide constituted together. So, liposaccharide is contained in the outer membrane. That being said, it means the lipopolysaccharide will only be found in the gram-negative and it will be absent in the gram-positive bacteria. I hope we are together till there. Now, one thing that you have to understand is that the lipopolysaccharide is what constitute one of the major toxins called the endotoxins. So that you have to have an understanding in. Now, if you go to the stain reaction, if you do a gram stain, the gram-positive tends to appear blue, or some will say purple. So that is the color that constitutes when you do a gram stain. On the other hand, the gram-negative bacteria will always appear pink or red. That's why this side appears in red. Let me just put this side as red in color, and all this side let it appear blue, just to make it more interesting. So if you do a gram stain, what happens? The gram-positive tends to appear blue or purple, and the other tends to appear pink or red. I believe that you can see clearly. Now, in terms of toxins, I told you guys that the gram-negative always contain the outer membrane. The outer membrane is always constituted with polysaccharide, and polysaccharides forms the endotoxins. So that being said, endotoxin is only found in gram-negative bacteria. So here we will see endotoxins, and also they also contain exotoxins. Yeah. So endotoxins and exotoxins will be realized in the gram-negative, whereas in gram-positive, because they lack the lipopolysaccharide on the outer membrane, they only have the exotoxins. Now, let me give a brief explanation on what I mean. There's something called toxins, and you have got two broad categories of toxins. You have got the exotoxin and the endotoxin. The exotoxins, basically, these are toxic proteins that are always secreted by living bacteria. Yeah, mostly gram positive, but and some gram negative. Yeah, so they're always secreted by living cells, and they majorly constitutes either proteins or enzymes, whereas the endotoxins are always part of the outer membrane of the cell wall, and are only found in gram-negative bacteria. So they're always released if the upon the death of the cell, yeah, upon the death, when the cell dies, it releases the endotoxins, and the endotoxins are majorly made up of the lipopolysaccharide. Yeah, so that's what you have to know. Now, in terms of antibiotic susceptibility, you have to understand that the gram-negative bacteria tends to be more resistant due to the outer membrane. So here you can say that uh, the gram-negative are more resistant. Why? Very simple, because they have outer they have outer membrane. They have got outer membrane. That's why they are more resistant. Whereas this one will be more susceptible, especially to a drug category called the penicillins. They are more susceptible. Next, if you look at the examples, I just give a major examples of the class that fall beyond that are found in the gram positive and the gram negative. Now, if you want to look at some example of the bacteria that are gram positive, very common, we've got the staph bacteria, the staphylococcus, you can say aureus, staphylococcus aureus, I think that's the spelling. Yeah, you've got others like staph pyogenes, so they're just many staph pyogenes. What are there's some so all the staph bacteria, they belong there. You can also have the strep like the streptococcus. Streptococcus, one of them is pneumoniae, very common. Pneumoniae, I think that's the spelling. So the streps, they belong to gram positive. They've got the enterococcus, also very common. A very common bacteria, especially amongst hospital acquired UTIs, very common. You also have got the rods, like the bacillus, very common. Bacillus is another common example of a gram positive bacteria, very common in causing anthrax. Let me just add here anthracis to be more specific. Very common bacteria that causes anthrax. We also have got the Clostridium. Yes, the Clostridium is another example. The Clostridium tetani. Yeah, I think that's the tetani. I think that's the spelling. Very common in causing tetanus, and the others that I don't want to discuss right now. So if you look at the gram positive, just know the staph, Genii staph, the strep, the enterococcus, the bacillus, and the Clostridium. All these are example of the gram positive bacteria. In gram negative, here one of the major common one is the Neisseria. Neisseria, you've got two groups. You've got the meningitis, which causes meningitis. You've got the Neisseria gonorrhea, which causes gonorrhea. Very common. We also have the E. coli, yeah, or Escherichia coli, another common bacteria that causes UTI, very common, and other diarrheas, illnesses. You also have got another one called Klebsiella. Sorry, very hard terms. Klebsiella pneumoniae and others. We also have got the Pseudomonas. Pseudomonas is another bacteria like Pseudomonas aeruginosa. Sorry to say, a very common bacteria. Others are Salmonella, typhi, like the one that causes typhoid, and others. I don't know to dig deep on major ones, but just have a clear understanding on some of the common bacteria that fall under gram positive or gram negative. Yeah, aeruginosa, what's the spelling? Aeruginosa, I think that's the spelling. So this is just a summary of the some of the bacteria that you have to know. So in short, a summary, for you to understand antibiotics, you have to understand microbiology of the bacteria. You have to understand bacteriology. And one key component or one concept that you have to understand, number one, you have to understand the cell wall of the bacteria, which is majorly composed of peptidoglycan layers. You have to understand the cell membrane. Yeah, one contains an outer membrane, and that's only the gram-negative bacteria. We also need to understand the nuclear composition, the DNA structure, the RNAs, the protein synthesis, how the DNA is being manufactured, the folic acid pathways. Once you have this understanding, then when we come to explain how the antibiotic work, it will become extremely easy for you to understand. So that's what I want you to understand. Then other thing or other concept, you have to understand the two major classifications of bacteria, the gram positive and the gram negative. The gram positive tends to appear blue or purple in the stain reaction. That's why in my table here, I have put this side in blue. In gram negative tend to appear pink and red. That's why in my slide here, I have got this column appearing in red. So once you understand these differences between the gram positive and the gram negative, it will become easier for you to understand how the antibiotic work. I think till there we have